Hello everybody. Today we talk again about TRANSFAC and how to use it to find composite modules. So compositions of binding sites for transcription factors.
Hello everybody, my name is Alexander Kell. And today we'll talk about using TRANSFAC to find composite modules. What are the composite modules? These are combinations of transcription factor binding sites in promoters or enhancers of genes that control their expression in specific conditions. For instance, in tissue, cell types, under signal transduction events in different cell cycle phases. So let's have a look. There are a number of good examples of composite elements documented in the TRANSCOMPEL section of TRANSFAC database. For instance, this example shows a chicken embryonic alpha-gobin gene, promoter of that gene, where there are two sites for SP1 and NFY, which binds to their neighboring sites, interact with each other, and activate the gene in primitive erythroid cells. In another type of cells, in a near site, another factor, which is NFY, binds there and represses the gene in all other adult cells. Another example in the rat serum amyloid A1 gene, where there are two sites for CBP and NF-kappa-B, which sit together in the neighboring sites and interact with each other and activate the gene in liver cells during acute phase response. But in another cells, the same region is occupied by YY1 factor, which remove those factors from their place and repress the genes. So it actually acts like that. On the same region of promoter, they can be in a silent phase of the gene. There might be a repressor sitting, which repress the gene. But once other factors which are coming and they activate it, and then they remove those repressors from that region and actually start binding to that region in synergistic manner. They cooperate with each other, form so-called enhanceosome, and they activate the gene. In our algorithm to search for the composite modules, we do like that. We take a promoter of that gene, and we search for transcription factor binding sites, which you can see here. And then we take this function, a normal function, and basically push it through the gene like that, and find the place where the most of the sites are located. And then we compute the score of that gene or that promoter, which is basically a sum of the scores of all the sites concentrated in that region multiplied by the weighting function which we push to the gene. At the end, we calculate the score for each of the promoter of the upregulated genes. And then we put these scores on this histogram. As you can see, we put the score for the upregulated genes here, the red guys, and the score for the non-changed genes here, the blue guys. And then our algorithm searches for such combinations of motifs that discriminate maximally the upregulated genes from the downregulated genes. And at the end, we build this model, which has these motifs and their combinations and the distances between them, and basically the score of the cutoff of the score of each of the motif. We will demonstrate how this search for the composite model works on this example of taking from gene expression omnibus on RNA sequencing of tumor infiltrating immune cells across 12 different types of tumor. And our interest will be focused on so-called Tregs, T regulatory cells, which are very important targets for tumor therapy, and we will compare them to the regular T cells. Regulatory T cells. Regulatory T cells are specialized subpopulation of T cells that act to suppress immune response by inhibiting T cell proliferation and cytokine production. They play a critical role in autoimmune diseases and cancer. High level of Treg cells in tumor microenvironment are associated with poor prognosis in many cancers. FOXP3 is a master transcription factor of Tregs. Upregulation of FOXP3 gene is required for Treg cell development. And basically we'll go to this entry and we'll download this original data on gene counts in our computer, and we'll put it, we have put them in this Excel file where you see all the genes and all the samples, and then we have here the values of measurements of expression of those genes done by RNA-seq. By yellow, you see here the data coming from T cells, and there are around 200 samples for T cells, and around 150 samples coming from Treg, shown here in blue. So we are going to analyze this data. So we load them into the geneXplain platform. First we log in, give our account. We are here in the platform. We go to our project. And first we have to do is to create a folder. So we create a folder, Treg versus T cell. Here it is. We go to that folder. And now we are going to load this data into that folder. So we find our file, Excel file. Click Start. Now it recognizes the format. So, and it's loading into the platform. Now it is here in the platform. You see the columns, the genes. Next, we want to find differentially expressed genes. We start Lima program, and we select this file. We select the entries, basically the columns which correspond to Tregs, and we select the columns that correspond to T cells. And the Lima will compare the values, the expression values in these samples between each other and compute basically the log-fold change. We will see it in a moment, and the p-values. Here it is. So we have the whole list of genes with log-fold changes and p-values. Now, next is to select upregulated genes. So here in the filter region, so we select the log-fold change should be higher than 1.5 and adjusted p-value lower than 0.01. Now we get this list of upregulated genes. We call the file with this list as up, and FOXP3 is a top upregulated gene. So now we have this entry here as upregulated genes. The same basically we do for the downregulated genes, where we select the log-fold change below minus 1.5, and the most important, we select the non-change genes. For that, we find those genes whose log-fold change is around the zero. So it's basically more than minus 0.02, but below 0.02. And we require that the genes are actually expressed. Their values of expression is higher than 5. So now we have those non-change genes. We save them as a separate file. So we have now files for upregulated genes, for downregulated genes, and for non-change genes. So what to do next? So we want to search for transcription factor binding sites. And for that, we go to our list where we have our pipelines. And we initiate the pipeline to analyze sites. We put here upregulated genes and non-change genes. We select the species human and the region of the promoter from minus 1000 to 100. And now you see how the pipeline works. So basically you see also how the reports of the pipeline is outputted in the program. At the end, the program returns you a report where you see all the motifs which were found, these positional weight matrices. You see also the list of these matrices. You see where the sites are found in each of the promoter of those genes which we analyzed. So in the upregulated genes, you can click on each of the site and see where exactly it is, what is the score of that site, and what is the motif actually. So now we look also in the list of transcription factors which were found enriched. And we, for instance, can search for FOX transcription factors. So here it is. We put the FOX as a search term. And you see here a number of FOX factors which were found here, whose sites were found to be enriched. And especially here you can see the FOXP3. So now we go into enrichment table and search for FOXP matrices. And we see here there are two matrices, FOXP3 and FOXP1. But both of them actually can recognize binding sites for FOXP3 sites. So we report which sites in which genes are corresponding to FOXP3 or FOXP1 matrix. And we can also filter that this gene contains at least one hit. So here you get 95 genes which have at least one hit for FOXP3 or FOXP1 matrix. So these are the potential target genes for FOXP3. Now next we want to actually generate binding sites in this promoters of these 95 genes, so in the selected genes. Again, we start the site search, and now we select not the whole list of upregulated genes, but these target genes for FOXP3. And now we have a frequency matrix, the enrichment matrix, in this smaller subset of genes, which actually targets for FOXP3. And now we will search for the composite modules. We run this composite module analyst. We put there all the sites we found in these 95 genes, in the promoters of 95 genes. And we specify the algorithm parameters. And most important, we choose this FOXP3 and FOXP1 as a focused matrices. Now here we select how many modules would be searched in this composite model. So minimum 4, maximum 8. So each module contains just 2 matrices, and the distances between sites should be up to 500. So now we specify the output, and we run this composite module analyst. So you see the run of that program, and now you will see the results. So you have these 95 promoters, and you see the sites in the promoters, and most important here down, you see how this promoter model works. So you see that the AUC actually is very high, 0.95, which is very good. And the model itself is here. So you see that it found 4 pairs, 4 individual modules in that model. For example, the first one is FOXP1 and SMAD3. Here you see actually the distance is up to 466, and there are also cutoff parameters. Let's have a look at the FOXP3 entry in our human PSD database. What is known about FOXP3? It's actually expressed in these T cells, T regulatory cells. It also demonstrated and reported number of known binding sites for various transcription factors, and among them AP1, for instance. But very interesting, if you look at the ChIP-seq data in the promoters of FOXP3 gene, we see SMAD3 binding sites. So which gives actually us an interesting insight that SMAD3 and FOXP3 are indeed connected. Let's have a look at SMAD3 matrix in the database, and we see it's connected to SMAD3 gene, so we can have a look through the gene. We see it's connected to relevant diseases, and we also see it's expressed actually in immune cells. We can see also various binding sites documented in the promoters of that gene. But most interesting, if you look at the pathways where these genes is involved, SMAD3, and especially in protein-protein interaction data, and search for FOX, we see there are FOX proteins where it forms various complexes. We can look also in the Google, and in this paper, it was actually shown that SMAD2 and SMAD3 are indeed interacting with FOXP3, and as it is shown here, that it's indicated that they require for FOXP3 induction. So this way we can see that our finding is indeed confirmed by literature. Let's have a look at the next module, FOXP3 TBX2. This module is shorter, 77 base pairs between the binding sites. And if you look at the database about TBX2 protein and gene, we see that it's actually expressed in the relevant tissue. And if you look at the promoter of that gene, there are a number of binding sites, and especially is ChIP-seq data confirms and shows that there is a FOXP1 binding sites in the promoter of that gene. If we Google TBX21 and FOXP3, we see that these two genes are epigenetically stabilized in Tregs. So that's confirmed the interactions indeed. So now let's have a look at another transcription factors which are found here in this composite model, snail and GATA3. So as for the GATA3, let's Google it, GATA3 FOXP3. We can find this paper which shows clearly GATA3 controls expression of FOXP3. As for snail is indeed very important for cancer. It is expressed in the tissues, and in the promoter of snail gene you see SMAD2, SMAD3 binding, FOXM binding, and in ChIP-seq data you see also FOXP1 and FOXP3. And especially interesting that this gene is involved indeed in cancer pathology, and if we Google snail together with FOXP3, in this paper it's clearly showing that expression and activation of FOXP3 is induced when activating by snail1. So now let's have a look at those genes. So 95 genes which we found which have binding sites for FOXP3 in their promoters. Let's just map them to gene ontology and check which gene ontology terms are enriched among those genes. And the first thing we see, these 3 genes which are actually negatively regulating interleukin-12 production. Why interleukin-12? Because actually it's known that interleukin-12 and FOXP3 are in negative relations. So actually interleukin-12 negatively control the FOXP3. And if you look at these genes, the top one is this gene called FLAM-F1. It's a very relevant gene in T-cell pathology, and also it expressed in the tissue and very specific. Now we see this gene is very important, and we also see this combination of binding sites which we found in the promoter. So FOXP3 gene is regulated by all these factors, so that makes the hypothesis about the physical interaction between these factors very plausible. In summary, we analyzed regulatory T cells. We took RNA-seq data, we put it in Excel file. And then we put this Excel file in our platform and start analyzing promoters of upregulated genes, searching for clusters for binding sites. As a result, we actually created a model which consists of pairs of sites, including FOXP3, GATA3, snail, and other factors which discriminate upregulated promoters and non-changed genes. And at the end, we applied this model to this one gene and many genes actually, and see what are the clusters of sites which drive this gene expression in Tregs. So with that, how to use this technique to, for instance, create artificial promoters? Watch next video.